Hello and welcome to my YouTube channel Anatomy Mentor. Today I am going to discuss a very important topic of neuroanatomy, which is the ventricular system and the cerebrospinal fluid of the brain. Now this topic is essential for medical students when it comes to the examinations and also considering the clinical relevance. This is an important topic. Today I will be discussing this in connection with the following learning objectives, in which I will talk about the anatomical organization of the ventricular system of the brain and explain the boundaries of each ventricle along with the choroid plexus. Also, I'll discuss the formation, circulation, and absorption of CSF. I'll be defining arachnoid villi and explaining their role in absorption of CSF. And also at the end, regarding the clinical application, I will be talking a little about hydrocephalus. Okay, so let's start with today's topic. What is the basic layout of the ventricular system? Well, there are 4 CSF-filled cavities lying within the brain, and out of these, 2 are the lateral ventricles. One is known as the third ventricle, and then there is the fourth ventricle. Also, remember, there is a small terminal ventricle which is present in the spinal cord. Now, here we have the cerebral hemispheres, and we can see that over here, this here is the shaded part, which is showing us in the grayish color the presence of the ventricular system within the cerebrum. So let's look at these here 2 paired C-shaped lateral ventricles. If we look at each lateral ventricle, it has this here forward projection lying within frontal lobe, which is called the anterior horn or the frontal horn of the lateral ventricle, whereas this portion here is the body of the lateral ventricle, which is present in the parietal lobe of cerebral hemisphere. And posteriorly, there is a projection of the lateral ventricle known as the posterior horn present in the occipital lobe. And here is the inferior horn, which is the projection of our lateral ventricle lying within the temporal lobe of the cerebral hemisphere. So this is a C-shaped lateral ventricle of one cerebral hemisphere, and here is a C-shaped lateral ventricle of the other cerebral hemisphere. Both these lateral ventricles, they are containing the cerebrospinal fluid, which then enters through these 2 communications known as interventricular foramina into a single median cavity, which is known as the third ventricle. From here, the fluid then traverses through a narrow passageway known as the cerebral aqueduct of Sylvius, and then this fluid enters into this tent-shaped cavity, which is the fourth ventricle. And remember, this fourth ventricle is present between the cerebellum posteriorly and the pons and medulla oblongata anteriorly. So these are our ventricles. They are containing cerebrospinal fluid, and they're lined by an epithelial layer, which is known as the ependyma. Okay, so here is a closer view of the ventricular system, and here you can see this is the C-shaped lateral ventricle of one cerebral hemisphere, and here is the lateral ventricle of the other cerebral hemisphere. And over here, B is the yellow arrow pointing towards the communication point of the lateral ventricle with the third ventricle. And like I told you, this communication point is known as the interventricular foramen of Monro. And from this third ventricle, there is the communication which is the cerebral aqueduct of Sylvius. And here E is the fourth ventricle. Now let's talk about the gross anatomy of the lateral ventricles first. So lateral ventricles are the 2 cavities, one within each cerebral hemisphere. And here we can see the central part of a single lateral ventricle, which is called the body. And the forward projection is anterior horn. Its posterior projection is posterior horn. And the lower downward projection is the inferior horn. The junction point of body with the posterior and inferior horns is the most dilated part of the lateral ventricle, which is called the atrium or the trigone. Now, let's first look at the relations of the body of the lateral ventricle. If we were to obtain a coronal section, then each lateral ventricle in the region of the body will appear to be triangular, and it will consist of a roof, a floor, and a medial wall. So here is a coronal section through the region of the body of the lateral ventricle. This here you can see is that region in which the section has been taken through the body of the lateral ventricle, the left and the right. As we know, they are paired. So like I told you, each lateral ventricle has a roof, a floor, and a medial wall. In the roof, remember, is present the corpus callosum. So these are the bundle of the nerve fibers connecting the two cerebral hemispheres to one another. So this here corpus callosum is present in the roof of this lateral ventricle. Whereas the medial wall of the lateral ventricle is formed by the septum pellucidum and also the fornix. And remember, this medial wall is common for both the lateral ventricles. And within the floor of the lateral ventricle, we can see medially is present the thalamus and laterally is the body of the caudate nucleus. And also present in the floor between the thalamus and the caudate nucleus are the stria terminalis laterally and thalamus striatum medially. So these are the relations of the body of the lateral ventricle. Also remember, the point between the fornix and the thalamus is the choroid fissure, where there is projection of the choroid plexus of the lateral ventricle. Now that we're talking about choroid plexus, first we want to know what is tilia choroidea. Remember that tilia choroidea is the double-layered fold of pia mater, which is lying between fornix superiorly and thalamus inferiorly. So if we go back over here to this diagram, we can see that this here is the tilia choroidea of the third ventricle. And so this is the double-layered fold of pia mater. And from the tilia choroidea, there is projection of the choroid plexuses. The choroid plexus is simply a vascular fringe of pia mater covered by ependymal lining. It is the irregular lateral edge of tilia choroidea which is projecting onto the medial aspect of the lateral ventricle. So here, if this is the tilia choroidea, you can see here is the projection, the vascular fringe of pia mater, which is projecting into lateral ventricle, known as choroid plexus. Choroid plexus, you know, is the site of production of the cerebrospinal fluid. Okay, so the blood supply of the tilia choroidea and therefore also of the choroid plexuses of both the third and lateral ventricles is derived from the choroidal branches of internal carotid and basilar arteries. So here we can see this is the, then again, this is a coronal section through the third and lateral ventricles. These are the lateral ventricles, the paired ones, and here is the midline cavity of the third ventricle. Now, as we have taken a coronal section, we can see here is the highly vascular tilia choroidea, and projecting from this tilia choroidea are the these are the choroid plexuses of the lateral ventricles, and here you can see are the choroid plexuses of the third ventricle. Two vascular fringes, they hang down from the roof of the third ventricle to form its choroid plexuses. And the choroid plexus of this third ventricle, it extends into the body of each lateral ventricle through the choroidal fissure. This point is known as the choroidal fissure. It is the C-shaped slit between the body of fornix and superior surface of thalamus, and it forms the medial wall of lateral ventricle. Also, it is the attachment site of its choroid plexus, and its anterior limit, remember, is the interventricular foramen. So here, let's take a closer view, a closer look at the choroid plexus. We can see over here through the coronal section of hippocampus and related structures, this red arrow is pointing towards the site of the choroid fissure. Choroid fissure, remember, is the point where the vascular fringe is going to be, that is, the choroid plexus is going to be projecting into the lateral ventricle. So this area, this choroidal fissure, it is bounded above by the tail of the caudate nucleus and below by the hippocampus. And the hippocampus is covered by a sheet of white matter, which is known as alveus, which then converges to form the ridge known as the fimbria. Now that we have discussed the relations of the body of the lateral ventricle, we are going to discuss relations of the anterior horn of lateral ventricle. If we were to take a coronal section, it also will have a roof, floor, and medial wall, just like the body. So its relations will be similar to the body or the central part of the lateral ventricle. But keep in mind that teliochoroidea and choroid plexus do not extend into the anterior horn of the lateral ventricle. So here is a coronal section through its anterior horn, which is present in the frontal lobe, and we can see that above is present the corpus callosum, medially is the septum pellucidum, and also the rostrum of corpus callosum. And you can see over here that this is the floor, and the floor, it has the chordate nucleus. So these are the relations of the anterior horn of the lateral ventricle. Now let's look at the relations of the posterior horn of the lateral ventricle. Over here, remember that there is a roof, a lateral, and a medial wall. So here is the roof and the lateral wall, and that over there is present the tapetum. Now, what is the tapetum? This is simply the bundle of nerve fibers that extends from the splenium of the corpus callosum. And you can see over here, this here is the cavity of the posterior horn of lateral ventricle. And if we look at its floor, there will be two elevations in the floor. The uppermost one is known as the bulb of posterior horn. This elevation is produced due to the fibers of forceps major. This is the bundle of fibers which extends posteriorly from the splenium of corpus callosum. And here you can see the other elevation, which is known as calcar avis. Well, calcar avis is the bulge which is produced due to the pushing of the white matter by the calcarine sulcus. So these two bulbs or elevations are seen in the floor of the posterior horn of the lateral ventricle. And you can also see that in the prosected specimen, the brain specimen over here, you can see these projections in the posterior horn of the lateral ventricle, the bulb of posterior horn, and the calcar avis, which is produced due to calcarine sulcus. Now coming to the relations of the inferior horn of lateral ventricle, remember the choroid plexus extends into the inferior horn through the choroid fissure. And the cavity of the inferior horn is very narrow on cross section, bounded laterally by the roof, and medially there is a floor. And also keep in mind that here the floor of the body of the lateral ventricle is going to be continuous with the roof of the inferior horn of the lateral ventricle. So like I said, the inferior horn has a roof laterally and a floor medially. So here is the roof laterally, and the structures present in its roof are the stria terminalis, tail of chordate nucleus, and the tapetum. Whereas in the floor we see are the hippocampus and the fimbria. Over here in the coronal section of the brain, we see here is the body of the lateral ventricle, and here is the inferior horn of the lateral ventricle. So keep in mind the junction of the body of the lateral ventricle and the inferior horn. The choroid plexus will be continued into the inferior horn, and it is going to project through the choroid fissure. In this prosected specimen, we can see the white arrow is pointing towards the choroid fissure, and this was the similar diagram that I had shown you before. Here is the prosected specimen. It is showing us the choroid fissure, and the red arrow is showing us the fimbria, whereas the black arrow is pointing towards, you can see over here, the hippocampus. So this is the point between fornix and thalamus where the choroid fissure is going to be present, and this is the point which is the site of attachment of the choroid plexus. Okay, now we're going to be talking about the third ventricle. Since we have discussed the gross anatomy of lateral ventricle, let's talk about the third ventricle. Like I mentioned before, this is a slit-like median cavity present between the two thalami, and it is communicating above with the lateral ventricles through interventricular foramina of Monro, whereas below with the fourth ventricle through the aqueduct of Sylvius. Now, what are the boundaries of the third ventricle? This has two lateral walls, an anterior wall, a posterior wall, a roof, and a floor. The lateral walls, each lateral wall is marked by a hypothalamic sulcus. The thalamus lies above this sulcus, whereas hypothalamus lies below it. And these two thalami, remember, are communicating with each other through an interthalamic adhesion, which passes through the third ventricle. Now, The anterior wall of third ventricle is formed by lamina terminalis, anterior commissure, and the anterior column of fornix, whereas the posterior wall is formed by pineal body, posterior commissure, and habenular commissure. The floor is formed by optic chiasma, tubercinarium, mammillary bodies, infundibulum, posterior perforated substance, and the tegmentum of midbrain, whereas the roof is formed by tilia choroidea, and from there two choroid plexuses hang down. So let's look at a simple diagram. This is a sagittal section through the lower part of the brain, and this is the area of the third ventricle. And its lateral wall, you can see, is formed by hypothalamic sulcus. Above it is the thalamus, below is the hypothalamus, and here is the interthalamic adhesion, which is the connection between the two thalami that passes through the third ventricular cavity. Also, we can see over here that in the floor are the mammillary bodies, and this is the region of posterior perforated substance, and also you can see the infundibulum and the optic chiasma. Anteriorly is the lamina terminalis, anterior commissure, and anterior column of fornix. Posteriorly is the habenular commissure and the posterior commissure and the pineal body. So these are the boundaries of third ventricle. Also in association with third ventricle are certain extensions which are known as recesses. You can see over here, this is again a sagittal section. You can see the infundibular recess extends towards into the infundibulum. The optic recess of the third ventricle is present above the optic chiasma. Here is the pineal recess, which is the extension of the third ventricle between the superior inferior laminae of the stalk of the pineal gland. And above that, above the pineal body, is the suprapineal recess. So these are the recesses or the extensions, you could say, of the third ventricle. Now we're going to be talking about the gross anatomy of the fourth ventricle. Now this is a tent-shaped cavity which is lying anterior to cerebellum and posterior to the pons and upper part of medulla oblongata. Above it is continuous with the third ventricle through aqueductus sylvius. Below it is continuous with the central canal of the medulla oblongata and spinal cord. It has two lateral boundaries, a roof, and a diamond-shaped floor, also known as rhomboid fossa. So here is a sagittal section. It's a simple diagram through the brain stem. It is showing this area, which is fourth ventricle, lying behind pons and upper part of medulla and in front of the cerebellum. We can see it is a tent-shaped cavity. The upper folds of this tent are formed by a sheet known as superior medullary velum and inferiorly by inferior medullary velum. And here is the nodule of the cerebellum. So roof superiorly is formed by medial borders, remember, of two superior cerebellar peduncles and a connecting sheet of white matter between them, which is known as superior medullary velum. So here you can see the superior medullary velum is going to merge with the white matter of the cerebellum. Inferiorly, there is a thin sheet which is devoid of nervous tissue. It is composed simply of ependyma and pia mater, and it is known as the inferior medullary velum. Remember, the fourth ventricle is communicating with the subarachnoid space. The lower part of the roof of fourth ventricle is pierced in the midline by a large opening known as the median aperture or the foramen of Magendie, whereas lateral recesses extend laterally around the medulla and they open anteriorly as the foramen of Luschka. Now, what about the choroid plexus of the fourth ventricle? This is a T-shaped plexus, and the vertical part of that T is double. It is hanging or suspended from the inferior part of the roof of the fourth ventricle, and blood supply to this plexus is by the postero-inferior cerebellar arteries. So now we're looking at the posterior view of the roof of the fourth ventricle after having removed the cerebellum, and we can see here is its choroid plexus. And you can see the vertical part of the choroid plexus. There is a double T, and here we can see is the median aperture, that was the foramen of Magendie, and here are the two lateral apertures, the foramen of Luschka. These are the openings of the cavity of the fourth ventricle into the subarachnoid space. You can see that this is present behind the pons and the medulla. Now, the lateral walls of the fourth ventricle are formed cranially by superior cerebellar peduncles, and caudally by inferior cerebellar peduncles. And the floor, as I told you, is the posterior surface of pons and upper part of medulla oblongata. So now we're looking at the floor of this here fourth ventricle, which is, you can see, diamond shaped. So it's also known as the rhomboid fossa. And this is present as this is the posterior aspect of the pons and the upper aspect of the medulla. They are forming the floor of the fourth ventricle. And in the midline is a sulcus known as the median sulcus. And on either side of the sulcus is a prominence or an elevation known as median eminence. This on median eminence, it is more prominent and bulging in the lower part, and this part known as facial colliculus. Beneath it is present the nerve fibers of which are emerging from the motor nucleus of facial nerve and forming a loop around the abducens nucleus. So that is the facial colliculus. On the lateral side of the median eminence is the you can see another sulcus, which is known as the sulcus limitans. This sulcus limitans, on its outer or lateral side, is the area which is known as vestibular area, and present beneath it are the vestibular nuclei. Also keep in mind the upper part of sulcus limitans, you have a bluish grayish area where we have these neurons which are containing melanin pigment. These cluster of nerve cells are known as substantia ferruginea, and this area is locus ceruleus. This is having, you could say it is a part of the reticular formation, so its function is related to wakefulness. Now let's look over here, the lower part of our rhomboid fossa. We can see extending from the median sulcus are these strands of nerve fibers called stria medullaris, and they are projecting laterally towards the inferior cerebellar peduncle. Looking again at the posterior view of the fourth ventricle, the cavity of the floor of the fourth ventricle, if we look at the lower part of this rhomboid fossa, we can see that there is an extension of the lower aspect of our sulcus limitans to form a depression known as inferior fovea. And this inferior fovea, medial to it lies hypoglossal triangle, lateral to it is the vagal triangle. Beneath the hypoglossal triangle is the hypoglossal nucleus, and beneath the vagal triangle is the vagal nucleus. And also you can see over here that between the vagal triangle and the gracile tubercle is this here area postrema, whereas the inferior angle of rhomboid fossa, there is a fold which is known as the obex. So now that we have discussed the gross anatomy of the ventricles present in the brain, we are going to be looking at these ventricles in a three-dimensional digital model, so we can get a better concept of the layout and the anatomy of the ventricular system of the brain. Okay, so now we are looking at a three-dimensional digital model of the ventricular system of the brain. And over here, we can see that this here is the large size lateral ventricle. It is a C-shaped ventricle, and we can see that it has different parts. The anterior extension, labeled number 1 over here, is the anterior horn that is present in the frontal lobe of the cerebral hemisphere. This here is the central part of body, which is lying in the parietal lobe of cerebral hemisphere. This here is the posterior horn, which extends into the occipital lobe of the cerebral hemisphere, which is labeled 2. And number 3 is the inferior horn of the lateral ventricle that extends into the temporal lobe of the cerebral hemisphere. So this here is the C-shaped lateral ventricle, and you know that this lateral ventricle, it contains cerebrospinal fluid. It is lined by a thin epithelial layer, which is called the ependyma. And these are the paired structures. So there's one lateral ventricle present in each cerebral hemisphere. As you can see over here, these are paired. Also, you can see the choroid plexus on the undersurface over here. The choroid plexus is the site of production of cerebrospinal fluid. It is present in the body of the lateral ventricle, and it also extends, as you can see over here, it also extends into the inferior horn of the lateral ventricle. Now, each lateral ventricle communicates through a foramen with this central single median cavity known as third ventricle. These sites of communication are known as the interventricular foramina of Monro. And when we see this here is the third ventricle, it also has its own choroid plexus hanging from the roof of the third ventricle. And the CSF, which is present over here, will then travel through the cerebral aqueduct of Sylvius, which is labeled 12. It's a narrow passageway, and it will enter the fourth ventricle cavity present over here. Let's look at the third ventricle. It has certain extensions, also known as recesses. Here is the infundibular recess and then the supraoptic recess, whereas posteriorly are the pineal and the suprapineal recesses of the third ventricle. This point 6 is showing the location of the interthalamic adhesion, which is the communication between the two thalami that passes through the third ventricle. So the third ventricle is sandwiched between the two thalami. And over here, if we look at the fourth ventricle, we can see that over here are the two location points on either side. There are the two lateral, you can see the two lateral recesses, and from there are present the two lateral foramina of Luschka. And over here in the median plane is the foramen of Magendie, that is the median aperture. Through these three openings, the CSF of the fourth ventricle is going to enter into the subarachnoid space. So this was a brief description of the ventricular system on a three-dimensional model, and I hope it helped you in getting a better understanding of the ventricular system of the brain. So now I am going to go back over here to our PowerPoint slides. And now we're going to discuss in detail the production, circulation, and absorption of cerebrospinal fluid. So here we're going to talk about that. We can see over here, now this is a diagram which is showing us, if you just follow the arrows, the point of production and the movement of CSF. So here are the black arrows. They are actually showing us where the CSF is being produced. So let's just zoom in and look over here that this is a sagittal section through the brain and brainstem, and this is the choroid plexus. You can see of the lateral and the third ventricles, and here the CSF is going to pass from lateral ventricles through the interventricular foramina of Monro into the third ventricle, and third ventricle over here, the fluid from there is going through aqueduct of Sylvius. This is present in the midbrain. It's a narrow passageway, and here the CSF has now entered the cavity of the fourth ventricle. And if we look over here, now we are going to follow the white arrows. They are showing the exit of CSF from the cavity of fourth ventricle through the lateral apertures of Luschka and median aperture of Magendie into the subarachnoid space. So now the white arrows are showing the circulation of CSF in the subarachnoid space. So this is the exit of the CSF from the ventricular cavities through the median aperture of Magendie. And here on either side of the medulla, you can see is the entry, or you could say the point of entrance of CSF through the lateral apertures. And now it is circulating in the subarachnoid space, and eventually it is going to be drained through these arachnoid villi into the venous system of the brain. So we're going to be talking about the drainage, and we're going to be talking about the arachnoid villi and granulations. Here is a basic layout. It is going to help us understand that CSF is produced, we know, by choroid plexus. The choroid plexuses are in the lateral ventricles, also in the third ventricle, as well as in the fourth ventricle. So the direction of flow of CSF is from lateral ventricles through foramen of Monro into third ventricle, then through aqueduct of Sylvius into fourth ventricle, And from fourth ventricle, remember the CSF also, it is going to enter the central canal of spinal cord as well and come back from there. And also it will go through the foramina of Luschka and Magendie into subarachnoid space. And as I showed you, from the subarachnoid space, it is drained into the dural venous sinuses, particularly this here superior sagittal sinus, which is the most prominent one present in the median plane. Okay, so now we are going to go ahead and we shall see over here an MRI to locate these foramina which were present in the fourth ventricle. The foramen of Magendie delivers CSF into different areas which are dilated areas where the CSF is present, and one of them, which is the largest one, is known as the cisterna magna, and that is the cerebellomedullary cistern. You can see the blue arrow over here, which is pointing towards the site of location, the location of the cisterna magna. So it is simply the CSF-filled space which is located within the posterior cranial fossa, dorsal to the medulla, and caudal to the cerebellum. So you can see this is the cerebellum, and just below it over here, and this is the location of our cisterna magna. Whereas the foramina of Luschka specifically deliver the CSF into the interpeduncular cistern. So there is another cistern, which is the cisterna ambiens. That is the connection between the quadrigeminal cistern and interpeduncular cistern as it extends laterally around the midbrain and thalami. So if we want to look at the cisterns, let's go back over here. We can see that these are the spaces over here. You can see this here is the cerebellomedullary cistern, which I just showed you in the MRI. That is the cisterna magna. It's a large space where the CSF is present. And also you can see this is the area of the quadrigeminal cistern, right? It is posterior to the midbrain. And we can see over here is the presence of, this is the location of interpeduncular cistern. So these are the different spaces which contain our CSF. Okay, so I was going to talk about the arachnoid villi granulations. So when we discuss the drainage of CSF, remember the drainage is at locations which contain the arachnoid villi. Now, what is an arachnoid villus? Structurally, it's a diverticulum, or you could say a prolongation or extension of subarachnoid space that pierces the dura mater, and it is covered by, you can see, a thin cellular layer and an endothelial lining of venous sinus. So what are the venous sinuses? They are simply the areas which are containing the blood, and they are present between the two layers of dura mater, that is the endosteal layer and the meningeal layer. So these are the spaces, and you can see this is a coronal section through the superior sagittal venous sinus. And here is the extension of that subarachnoid space, and it's projecting into the venous sinus. And this is a finger-like projection, so that's why it's called a villus, which literally means the shape of a finger. This is an arachnoid villus. Here are the pia mater, the arachnoid mater, and the dura mater. So this subarachnoid space is projecting into venous sinus. And where there are many arachnoid villi, those bunches of arachnoid villi are known as the granulations. Now, if we want to talk about the physical characteristics, the composition of CSF, then we should know that it's a clear and colorless fluid, and its total quantity or volume is about 150 ml within the ventricular system of the brain and spinal cord, and about 500 ml is produced daily, and that means that it is constantly going to be replaced, since it's going to be maintained at a volume of 150 ml. And about 30 ml of this 150 ml, approximately 25 to 30 ml, is present within the ventricles. Okay, and also when we look at the pressure that is produced because of the CSF, when we check that pressure, it is about 60 to 150 millimeter of water when checked with the position of the patient in lateral recumbent position. Considering composition, remember the protein content is minimal. The glucose is about half that of blood plasma, and there are a very few number of lymphocytes present within CSF. What are the functions of cerebrospinal fluid? It is a fluid cushion which protects the central nervous system, namely our brain and spinal cord, against mechanical trauma. And it also provides buoyancy and support for the brain. It provides nourishment of nervous tissue and removal of waste products. Now we're going to discuss in the end the clinical condition, which is hydrocephalus. So up till now, we've discussed the basic anatomy, the gross anatomy of the ventricular system, and now we're going to talk about an abnormality, which is hydrocephalus. Well, hydrocephalus, it is an abnormal increase in CSF, in the volume of CSF in the skull, and that can lead to an enlargement of the head in children, because in children we know that the sutures have not permanently fused. There is a level of compliance. There is therefore an expansion of the head in that age group if hydrocephalus, that is known as hydrocephalus, if the CSF pressure is raised. Although compensatory atrophy of brain tissue can also occur with the normal CSF pressure. So the raised CSF pressure, it can be due to increased production or some blockage in the circulation or decreased absorption. Now, we can classify hydrocephalus in typically these two types. One is obstructive and the other is communicating. As the name shows, the obstructive is due to an obstruction at some point in its circulation and its drainage, whereas in case of communicating hydrocephalus, there is no visible obstruction. Where the points of obstruction, where it will be most likely to occur, obviously the narrow passages such as interventricular foramina, aqueduct of Sylvius, and apertures of fourth ventricle, these are the sites vulnerable to obstruction. Dilatation will occur in all the cavities proximal to the obstruction. Now, it can also be caused, the obstruction, due to a tumor or inflammatory exudate secondary to conditions like meningitis. Decreased absorption of CSF, that can also be due to inflammatory exudate, venous thrombosis, pressure in the venous sinuses, or obstruction of the internal jugular vein. So here we can see that this is showing us the ventricular system, which has been enlarged due to the increased quantity or volume of CSF. And clinically, this can be assessed when in children as there is going to be a bulging or a prominence of the anterior fontanelle. And over here, the cause seems to be the blockage of the aqueduct of Sylvius. So you can see the cavities proximal to this blockage, those ventricles have been enlarged. And this can also cause pressure effects on the surrounding brain tissue. It is typically corrected surgically by the insertion of a shunt. This is known as the ventriculoperitoneal shunt, which is going to withdraw the fluid, the extra fluid, from the ventricular system into the peritoneal cavity. Okay, so now that we have covered the essentials of the ventricular system, let's just discuss a few MCQs which you should be able to answer now. The bulb of the posterior horn. Now we're talking about the posterior horn of the lateral ventricle. The bulb of the posterior horn, that is produced by the forceps major. And the roof of the inferior horn, that has the stria terminalis. The anterior wall of the third ventricle, as we discussed, over there is present the lamina terminalis. Okay, so that concludes with today's discussion on the ventricular system and the circulation and the absorption of cerebrospinal fluid. I tried to stick to the basic concepts, and I gave you an idea of the organization of this ventricular system, and also a concept which was based on the digital model, so you can understand also the relations of the ventricles. So if you found today's discussion helpful, don't forget to like and subscribe to my YouTube channel, Anatomy Mentor. You can simply do that by just pressing on my picture, which is going to appear at the end of this video. And also don't forget to press the bell icon so you can be regularly notified of any new updates, any new discussions on different topics of anatomy. Okay, so stick to your studies and stay blessed. Thank you. Take care.